muscles. And you're like, what the hell's wrong with the rest of you people? Okay, so I'm just going to throw that out there. If you are at all interested, ask the TAs that you have now. Most of them enjoy it, otherwise they wouldn't be doing it. Go ahead. 2327, it's instructor experiences in anatomy. It looks good on resumes and stuff too. Okay, so today we are going to be doing nervous tissue. And I am going to torture you by trying to make you use the words that you learned last time. So whenever possible, I'm going to ask you afferent, efferent, CNS, PNS, right? Motor, sensory, what is it actually doing? Somatic, autonomic, all of that kind of stuff. So you will only get familiar and confident with those words if you force yourself to use them and force yourself to know what you don't know about them. And they like really take some practice, honestly. So we're going to start with Roman numeral 3, which is nervous tissue. And I'm going to remind you that when you look at nervous tissue, especially if you're looking at the gray matter of nervous tissue, what you're going to see is, of course, neurons and glial cells, yes? Not all neurons look like this. We'll talk about what the other ones look like. Remember, hey, you guys, it's my turn to talk. Remember that primarily when you're looking at a slide like this, would you be looking at that gray matter or white matter? Gray matter. If you're seeing neurons, you're primarily looking at gray matter, right? White matter doesn't really have any neuron cell bodies in it, right? It's just cabling between them. So this would be white matter. Ooh, and I found you a cool picture. Sorry, that would be gray matter. This, what is this? What is that? Yeah, it's white matter. This is a nerve, right? It's a bunch of, and look, they took one of these from the sciatic nerve, and they did a cross-section, and you can look at it up close. Woo! That's super neat. This one is an axon, right? This one is going to be the myelin sheath, and what's going to be this? Try again. Keep going. Endo. Yes, that's the endoneurium. So that's a super close-up picture, right? That won a science photography contest last year. It's a pretty cool picture. I think it's awesome. Okay, so let's look at neurons, and then we'll look at glial cells, okay? So what is special about a neuron? So this is the histology slide, or close to the one that you studied before. It doesn't all look like this. This is oversimplistic, but let's talk about what neurons have in common with other cells and different with other cells. First off, neurons are incredibly energetically expensive, have a very, very high metabolic rate. They use boatloads of glucose and boatloads of oxygen, and they store almost zero glucose. Did you find it? Oh, good. Huh? Oh, good. Okay. They store almost, thank you for being trustworthy. Hope she didn't steal a cadaver. Do you think she did? No, she looks too clean. Okay. So they use tons and tons of oxygen and glucose. And this is really important because they don't store any oxygen and they need constant amounts of oxygen. So for instance, what would happen to the gray matter of my brain if I lost blood flow to a region of the gray matter of my brain? It would die. Yes, it is going to die because it stores almost zero oxygen. And remember that that's super important with this tissue because almost without exception, neurons are amitotic after fetal development, right? They are not going to regenerate. So if you kill off a neuron cell body, it's pretty much gone. And this is important too. So the fact that they're amitotic after fetal development, this is important for those of us who could have a pregnancy or have had a pregnancy, right? So you don't get any more neurons after fetal development to speak of, which means you get one shot to put together a nervous system. One, okay? And you start actually having differentiation and development of the first neurons about 3 to 5 weeks post-implantation. So you would be about 6 weeks pregnant when you're developing your first neurons, which is really, really important to know. So 6 weeks all the way to the 40th week, you're going to be developing neurons. Really important because you only get one shot to put together a nervous system. And if, for instance, you are not taking adequate care of yourself, you are not giving yourself adequate nutrients, you are not giving yourself adequate vitamins, you are, for instance, deciding this would be a good time to try crack, no, it's the worst time to try crack. Because this is the only time, I mean, you know, right? You thought, I thought it would be a good time to try crack. that it's really not ever a good time to try crack, but that's the worst time. So you don't ever regenerate any of the damage that occurs during that time, which is pretty freaking scary, right? Because a lot of times you don't even know you're pregnant, right? You're like, oh, I've been pregnant for ages. I probably should have eaten something other than pizza in the last 3 months. Go ahead.
So what about like diseases like neurofibromatosis and fibromyalgia?
Those are mostly functional diseases. Yes, spina bifida occurs embryonically.
Yeah.
And that is primarily not about cell division, right? But it's about the proper organization, right? So when we get to the next set of notes, we'll talk about spina bifida. And the other ones are not so much about the cells dying off, but the cells, not pathological anatomy, but pathological physiology, like how they are actually functioning. So we can talk a little bit about that in the next set of notes. So in addition to being amitotic after fetal development, that also means that they have to live as long as a person lives, yes? Which, I don't know, the oldest person the last time I looked it up was 123. What the hell are you going to do at 123? Like, who are you going to hang out with, really? Right? Everybody you've ever known is dead. So you don't keep all of them. You keep a lot of them, though. So the other things that are special about neurons is that they have two things that they do. They can receive almost any kind of stimulus, light, temperature, pressure, pain, chemicals, and they can convert it into an electrical stimulus called an action potential, right? And so muscle cells can do that too. It's incredibly energetically expensive. And then the other thing that neurons can do is that they can actually cause another cell to have an electrical stimulus because they can actually release neurotransmitters that will force you to have an electrical stimulus. And they're the only cell that can do that. They're the only cell that actually releases neurotransmitters. Okay, so I want to talk functionally about neurons now, and I want to talk about what they're doing and how to categorize them functionally. And we're going to start using terminology that you guys used last time. So if I look at a really simple example right here, I've got 3 neurons. I know that one of them looks weird, and I promise I will address that in just a second. Here's a neuron that is carrying sensory information in, okay? This woman had an itch on her skin, right? And she's going to respond by scratching it. That's what you're seeing here. So this neuron is carrying sensory information in. I'll tell you why it looks like that in just a second. This neuron is carrying information between 2 neurons. And this neuron is carrying information out to something that wiggles or squirts. Yes? Okay. So let's define these 3 types of neurons functionally by what they're doing. This neuron right here can be considered a sensory or what afferent neuron? Afferent neuron, yes. Okay. It's carrying information from a sensory receptor toward the central nervous system, right? That makes sense? This neuron right here is carrying information to a skeletal muscle fiber. So it is a motor or what afferent neuron? Efferent neuron, right? Okay. And then this neuron is neither sensory nor motor. It's between 2 neurons. It's in the central nervous system. It's called an interneuron because it's between neurons, yeah? Okay. And if it's not doing sensory or motor function, it's doing that CNS function. What's that CNS function that we talked about last time? Integration, yes? That's what it's doing? Yeah? So the interneuron is doing integration. Does that make sense? Okay. So if you look at a neuron and you see what it's attached to, you can tell whether it's sensory, motor, or an interneuron. Yeah? So now let's add just a little bit more information to these because you are capable of it after our discussion last time. And that is, now I want you to tell me specifically what kind of sensory neuron is this? It is carrying what kind of sensations? Yes, this is somatic sensory because it's coming from where? The skin. Yes? Okay. This is just interneuron and it doesn't get any fancy words associated with it. And this one is motor, but we can be more specific. What kind of motor is it? It's somatic motor, right? Because it's going to skeletal muscle, specifically skeletal muscle. Does that make sense to you guys? Okay. So we've got somatic sensory and somatic motor. Okay. So now that's telling what an individual neuron does functionally, right? Okay. And then now let's look at why the hell this one looks weird and talk about them structurally. So if we look at structural classifications of neurons, we can say that Although we initially sort of lied to you because that's half of teaching is figuring out when to lie and when to tell the rest of the truth, right? Tell the whole truth of anatomy in the first week, what happens? Everybody drops, right? No, we can't do that. We have to like little pieces, right? So the whole truth is that not all neurons look like this. This is one of the categories of neurons that we have. But there are some variations in neurons. These are the categories of neurons that you're going to run into. So you define these structurally by how many poles or processes they have sticking off of the cell body. You can have one process sticking off, you can have two, or you can have, I don't want to count them, okay? So let's look at each of these situations. This is the one that you've seen most often, right? This one is structurally called a multipolar neuron because it's got a bunch of processes sticking off of the cell. This one has how many processes sticking off? Just one, right? One attachment. And then it branches, right? But one attachment to the cell body. And how many does this one have? Two. Unipolar, bipolar, multipolar. Good? Okay, so now let's go back and do this one more time. So you guys told me about these structurally, and now you're going to tell me about them functionally, right? So functionally, this was somatic sensory, yes? Now structurally, tell me what kind of neuron that is. Unipolar. Now let's think about why the hell it makes sense to use a unipolar neuron there instead of a multipolar neuron there, okay? If I was doing sensation with a multipolar neuron, then my neuron cell body would be way out here in the periphery, right? Way out here. It's not very safe out there, is it? Right? So instead of doing that, I actually use a different kind of neuron that allows me to sense out here, but the neuron is all the way either at or near the central nervous system, right? Because that allows me to do that, doesn't it? Right? Do you see? So it allows me to put the neuron cell body either in or near the central nervous system. So you've got this unipolar one. So does it surprise you at all that lots and lots of sensory neurons are unipolar? Because then I can sense out here, because if you look at it, guys, look. These are dendrites, yeah? That's receiving information. But I actually start sending the information before it even goes by the cell body, right? If I use this kind, then the cell body has to be all the way out by the sensory receptor. Does that make sense? So this one allows for you to protect your sensory neurons far more. So I've got this situation right here in which the sensory neuron, that one is actually not in the CNS, but it's close to the CNS. It's in a little ganglion right near the CNS. Does that make sense? Okay. So you said that functionally, one more time, just hear the question. Functionally, that neuron is, be as specific as you can. Functionally. Somatic, exactly, somatic motor. And structurally, that neuron is unipolar. Okay, this one in the middle, same question. Structurally, that neuron is what? Structurally. Multipolar. Functionally, it is, no, it's an interneuron, right? Interneuron between neurons, that makes sense? Okay. Last one, bottom one, as specific as you can get it. Functionally, that neuron is somatic motor. Structurally, that neuron is multipolar. Okay. Now, you didn't see any bipolar ones. They're not very common. You find them, for instance, in the retina of the eye and a few other places. You just don't find them very often, so they don't fit into a lot of the pictures that you see. Does that make sense? Okay. All right. So now let's look at a multipolar neuron. By the way, multipolar neurons are by far the most abundant. You find them in the gray matter of the brain and the spinal cord, which is where most of your neurons are, right? Like just sheer numbers. It's mostly multipolar neurons, which is why when they teach you guys the first histology slide, it looks kind of like this, right? That's not what all of them look like, but it's certainly very common. So this is a multipolar neuron. So let's look at this multipolar neuron. Now, by looking at this picture, now everybody can identify that this is a multipolar neuron. Can you tell what this neuron in this picture is doing functionally? Is it sensory, motor, or interneuron? You can't tell because it's not attached to anything, right? So there's literally no way that you could figure out what it was doing unless you attached it to another neuron or a sensory receptor or a muscle, right? So you can only tell this one structurally. Does that make sense? Okay, so now if you look at a multipolar neuron, here's the histology slide and here's a diagram of the same thing. So here's a diagram of the same thing. And you've seen a diagram that looked like this, similar to this when you did the histology chapter. So we've got the soma or the cell body, right? And what's in the soma or the cell body? Cell stuff, right? It's like you've got a nucleus and a nucleolus and a bunch of other crap. Usually these look kind of grainy in their cytoplasm because they have boatloads of ribosomes, because most neurotransmitters are amino acid or peptide based. Does that make sense to you? Okay. Ribosomes do what? Protein synthesis, right? Using amino acids for protein synthesis. So if you look at that neuron and its histology slide, can you see how it looks kind of grainy? They call it chromatophilic substances. What it really is the rough ER and free-floating ribosomes that you're seeing there, because there's a lot of protein synthesis that they have to do. Is that all right? Everybody in anatomy gets pissed off. They're like, you are talking 1610 stuff, and I want you to shut up. But no, it's all the same subject, FYI. Okay? And in phys, ooh, they get super mad. Huh?
It is biology.
It is biology. In phys, they get mad when I'm like, hey, where does protein synthesis go on in the cell? And they're like, shut up. I'm done with 1610. It's like, come up with the word ribosome or else I will not stop staring at you. And I'm like, all right, ribosome. Okay, so now let's look at the processes that are, oh, sorry, one more thing. This is conceptually weird to talk about, but if you have a blob of neuron cell bodies, blob of them, what color does that tissue usually look? Great, yes, you got it, good. Okay, so I have a whole big blob on the outside of my brain. It's called the cerebral cortex, yes? I've also got some blobs deep within my brain. When I have blobs that are deep within the brain of neuron cell bodies in the brain and the spinal cord, do you see those that aren't on the surface, but it's gray matter on the inside? Yeah? Those are called nuclei. So it's a different use of the term nucleus. It is a blob of gray matter deep within the central nervous system tissue, okay? And we will start to figure out in the next set of notes, which is why you do not want to miss next Monday or Wednesday, because the brain is very hard to do by yourself, okay? We will start to figure out that each one of those damn individual blobs has a different function. They'll be like, this is the amygdaloid body. It's a nucleus within here, and it's responsible for rage and emotion. And you're like, what the hell are you talking about? You just said a whole bunch of words. So we can find it. We can do this together, but a lot of times you need a little guidance to do it. So blobs of neuron cell bodies within the central nervous system are called nuclei. Blobs of neuron cell bodies within the peripheral nervous system are rarer. Yes, there aren't as many of them. But when they exist, they are called ganglia. So let me show you that you actually have seen one, but you didn't know you were seeing it, and that is this guy right here. Okay, right here, neuron cell bodies. They're only showing you one, but I'm telling you that there's hundreds in there. That little blob right there that's right next to the central nervous system but isn't in it, that's called a ganglion. Okay? So I will reiterate that as we go through the next set of notes. Okay. Now let's talk about the processes that are attached to a multipolar neuron. Now, FYI, in this story that I'm telling, I'm talking about a multipolar neuron because dendrites and axons are easy to get confused when you're talking about unipolar and bipolar. So I'm going to talk about multipolar, and then we can go back and visit the other two and figure out how the hell does that work. So multipolar is actually pretty easy. With a multipolar neuron, you've already learned this. Dendrites receive information, but it's not magic that they receive. They receive information because they have receptors on them, cell membrane receptors, yes? And the primary type of information that they receive is chemicals. So you can like release chemicals and it binds to the receptor, right? And so that's why we've always said that dendrites are for receiving, right? And then the axon actually sends information out. And the reason it sends information out is because it has the physiological capability to make any stimulus into an electrical stimulus and send it 250 miles an hour down an axon. So dendrites are going to receive information. So if you looked at them really closely, right, you would see a whole bunch of cell membrane receptors on them. So although in this figure, you really can't tell the damn difference between a dendrite and an axon, right? They just labeled it and you're like, okay, right? doesn't really look dramatically different, the dendrites and the axons. They are physiologically different because the dendrites have boatloads of cell membrane receptors on them, and the axon has the capacity to generate an electrical stimulus because it's got these ion channels on it called voltage-gated ion channels. So although histologically they don't look different, functionally they're really different, okay? And you're taking phys, so you know what I'm talking about. She's like, oh yeah, they are. Okay, so now let's talk about axons. So let's look at this axon. Okay, so if you look at this kind of diagram, you can tell the dendrites from the axons, right? Because axons can be myelinated. So no matter how many dendrites you have, there will only ever be one axon, right? Axons carrying information away from the cell body. Now, it can branch as many times as it wants to, but there's still only one axon, one attachment to the cell body, right? It can branch as many times as it wants. So only one attachment. The attachment point right there where it attaches to the cell body is called the axon hillock. And physiologically, again, it doesn't look impressive anatomically, but physiologically it has a whole bunch of those voltage-gated channels on it and it's able to start an electrical stimulus, okay? So it's got the axon hillock. Now, so what we're going to do is figure out how it could branch. So if it branches in the middle, so here's my neuron cell body, right? Here's my axon, yeah? If I had a branch right here, it would be called an axon collateral. And you can have as many of those as you want, okay? And they don't always have them, but if they do, they can have as many as they want. They're called axon collaterals. What you always have, however, at the end is you always branch at the end. And these are called telodendria. Telo means end. Dendro means what? Fingers. Yeah, it means fingers. So it's literally the fingers at the end. Does that make sense? Okay. So these are axon telodendria. And then if you go all the way to the end, you are going to run into the fact that the fingertips do not look like mine. They look like ET fingers. You guys know what ET? I know you like 12, but the rest of you old people seen ET? Yeah? All right, cool. So, ET, what?
Smokers have it too.
They have,
They're like,
they do?
yeah, their fingers, but their fingers.
Oh,
So
interesting. Okay, so if you get to the end, the tips look really bulbous. And nobody can decide what to call those. They call them something different in every damn textbook. I don't know why the hell they didn't decide. They call them axon terminals or terminal boutons or synaptic terminals, whatever. They're the things at the end. Anybody know why they're bulbous like that? They store something. What do they store in there? Yes, they store neurotransmitters in there, right? That's where the neurotransmitters are going to be stored, and that's where they're going to be released from, okay? So those are the synaptic knobs, synaptic terminals, whatever the hell you want to call them. So they will release neurotransmitters, and the neurotransmitters are how you communicate with the next cell, the next nerve or muscle cell, okay? And we'll get to that in just a second. Okay, now some axons, but not all, have a myelin sheath. And we've talked briefly about the myelin sheath before. What the myelin sheath is a big fatty layer that goes on top of each neuron and axon, and it insulates the axon and allows that electrical stimulus to go very quickly down the axon. So the metaphor that you can use for it is like, okay, so you've got a wire, a copper wire, and you want it to like light up a light bulb. If you're doing a science experiment with your kid and you're like hooking it up to a potato and trying to light the damn light bulb up, and you can totally do that. Do you need to insulate the wire if it's really short? No. Now, if you want it to run through your house, you need to insulate the wire, not only because you can burn your damn house down, but also because if it touches something, you can actually lose the charge, right? So the longer an axon is, the more likely it has to be myelinated. Yes? So let's talk about how long axons can be again, and remind ourselves that an axon can be from the lumbar region of my spinal cord all the way down my sciatic nerve to my little toe. That is a really long axon, yeah? Okay? When axons are really long, they can be called nerve fibers. So let's reiterate what we talked about last time. You cannot call a neuron a nerve, yes? Ain't the same damn thing. An axon is a part of a neuron, right? If it's super duper long, like that one going through the sciatic nerve, it can be called a nerve fiber. So what we are seeing right here, again, is a collection of nerve fibers or axons, yes? And you call a collection of nerve fibers or axons a what? Nope. Collection of nerve fibers or axons. Wrapped in connective tissue. The whole thing is a nerve. Yes. Yeah? Call the whole thing. Does that make sense? Okay. So this is the whole thing is a nerve. So this is the terminology that people have trouble with. They use all the terms interchangeably, but if you practice, you'll get past that. Okay? All right, so let's talk about that myelin sheath and what it ends up doing. Okay, so the myelin sheath that we saw down here, what it really is this fatty coating around each axon. And the reason it's fatty is because what it really is another cell, a type of glial cell, devotes its entire self to wrapping around and around and around the neuron or the axon. And it wraps so tightly that it pushes everything, like all of the cytoplasm and the organelles and the nucleus, it pushes them out so that primarily what you have is layer after layer after layer of phospholipid. Does that make sense? Because the cell membrane is made of phospholipid. So let me show you how you actually do that. So the way that you actually myelinate is, so the yellow is the axon. Then you've got a cell. This is a specific type of glial cell that I'll talk about in just a second. And it devotes its entire cell life to wrapping around, and then it keeps going and wrapping around further until you have layer after layer after layer of phospholipid. So it's mostly fat, right? It's mostly fat. Now, wherever you have a lot of myelin, it tends to be white. So the tissue and the nerve tends to be white. And why is that? Well, if I have a single layer of saran wrap or cell membrane, it tends to be transparent. But is a whole roll of saran wrap transparent? No, it's opaque and usually kind of whitish, okay? So wherever you have a whole bunch of myelin, it usually makes the tissue look white, which is why nerves out here look white and why the white matter in the brain and the spinal cord looks white. Now, one more time, because we did this last time. What is the function of white matter in the brain, for instance? What's it doing? Huh? Connections, exactly. It's the wiring, right? So that's the metaphor that we were looking for, right? It's allowing for this blob of decision-making gray matter and this blob of decision-making gray matter to communicate with the wiring. Does that make sense? So wherever you have a whole bunch of myelin, it tends to be white, whether it's CNS or PNS. So if we look at this brain picture that we've got again, just look at this brain picture. You can tell gray matter from white matter, right? Wherever you have white matter, primarily what you're doing is not making decisions, but sending decisions, right? From one place to the other, okay? All right, go ahead.
So younger people don't have as much myelinated brain tissue, right?
Actually, you have more when you're teenagers and you actually start pruning it. So you actually have more synapses as a teenager than you do as an adult. So you have this weird little thing in which when you're really young, you don't have a whole lot of white matter, right? And then you have this bloom of white matter in which you start making connections to everything, right? And then as you go through your teenage years, part of what happens in the teenage years is you start pruning connections, which is an interesting thing to think about, like metaphorically, because like half of adolescence is like figuring out who the hell you are compared to everything you could be, right? Like, I don't like that music. I ain't gonna hang out with you, right? I think that's stupid. I like this, right? So what happens during that age, like it's like 14 to 19 primarily, is you have a dramatic pruning of all of the connections that you used to have. I totally see that with my 17-year-old now. He literally thinks everything is stupid. He's like, that's stupid, you're stupid, I hate that, that's the dumbest music ever, right? I'm like, what happened to you? You didn't used to be an asshole. And I was like, do you know that that's not a good way to make friends? Do you know that? He's like, I don't want to be friends with you, I don't like you. Okay, go ahead.
So if the axon is longer, is it the myelin thicker?
Yes,
If
it does usually have more insulation, and the axon's usually thicker as well. So it's actually counterintuitive. You have more white matter at that age than they'll have at my age. you tend to prune in sort of, I don't know if it like exactly goes with that metaphor, but it's an interesting overlap, right? We will talk about adolescent brain development when we get to the nervous system next. I have a phenomenal article for you guys to read that literally none of you guys read last time, the youngsters. You should read it because then you'll be one of the smarter youngsters, right? You're like, what the hell's going on with my brain and why do I hate everything, right? And only want to listen to my peers. That's part of what you're supposed to be doing. Go ahead.
So the axon is myelinated, but
Yes.
is the nerve?
No. So you can't say that the whole nerve is myelinated. You can say that each axon within the nerve is myelinated.
Okay.
Yes?
Yeah.
Okay, and then it's wrapped in connective tissue. But between all of that myelin and the connective tissue, it tends to appear white. Does that make sense? Okay, so now there's two different ways of forming myelin. They both accomplish the same thing, but you do it differently in the CNS. Oh, by the way, here's a picture, transmission electron micrograph picture of myelin, where you can actually see the layers, which is kind of cool. I like that one. Okay, so it looks like a what? Okay, so there's actually two different ways of myelinating an axon. The one that I just showed you was this one, right? Okay. There's another way of doing it. You myelinate differently in the CNS than in the PNS, which ends up having clinical significance. Okay. The bottom one looks like sausage links, right? Looks like sausage links. This is how you myelinate an axon in the PNS. For instance, this would be how my sciatic nerve is myelinated. Does that make sense? Okay. And the types of cells that do it are called neurolemocytes or Schwann cells. And what's important is that if you damage PNS, white matter, neurolemocytes or Schwann cells will try to help with repair. You don't have a guarantee of repair, but you have a possibility of repair if you damage white matter in the PNS. However, in the CNS, up here, they have a different form of myelination that ends up accomplishing the same thing, okay? So notice that instead of the cell devoting its entire little cell self to wrapping around, it sits off to the side and sends out fingers and myelinates a bunch of different axons, right? That makes sense? You're still getting your sausage links, they're just formed differently, yeah? So the cells that do myelination in the CNS, like in the white matter of the brain, right? These are called oligodendrocytes. Oligo means many, dendro means fingers, the many-fingered cell. Now why the hell do you care what type of myelination is done? Because if you damage white matter in the CNS, the brain or the spinal cord, you are screwed. Because oligodendrocytes do not help with repair. If they get damaged, they're like, I'm out, right? So, I bet you knew before you took this class that you didn't want to damage your brain or your spinal cord. But now I'm telling you that there are two reasons anatomically. Amitotic gray matter, yes, and also white matter that's not going to help you if you damage it. So it doesn't matter what you damage in your spinal cord. The damage is almost invariably permanent. Does that make sense? Okay. So the white matter you're never going to get back. Okay. So that's two different ways of doing myelination. PNS is neurolemocytes. CNS is oligodendrocytes. Okay. Notice that either way you're going to end up with these little bare spots in between. Those are called nodes, or neurofibril nodes, or nodes of Ranvier. It doesn't matter. If you hear the word nodes, that's what they're talking about. Okay? Those are electrically active, and when you take physiology, you will learn that that's actually like how you get things going really quickly, is that the nodes actually speed it up. Okay, so that is myelination and white matter. Okay, so now, the good part about learning anatomy of the nervous system is because it actually allows you to understand physiology of the nervous system pretty well. So now, just by looking at this picture right here, you could label dendrites, yes, and axon. How about the axon hillock? Could you label that? Axon hillock. You could tell me what structural category of neuron it is. Yes, what is it? Multipolar. Can you tell me what functional category it is? No, you don't have enough information. Can you tell just by looking at it which direction information is flowing?
Yes.
Yeah, you can tell that information is flowing this direction. Can you label the axon and the axon telodendria and the synaptic knobs, right? Okay, now this ends up being really important for phys and further neuro classes, because if you immediately look at the anatomy and you know the physiology, then you're a lot stronger, right? So now what we're going to do is we're going to take that and we're going to make it talk to another cell at the space that you talk between two cells is called, between two neurons is called a synapse. Okay, now look at this figure, even though it's slightly unfamiliar. Do you see that what you are seeing is the axon telodendria of one cell are coming into contact with the dendrites of the other one? FYI, they don't actually touch. They're actually microscopically separated with a little bit of interstitial fluid between it. And that's important because you actually release chemicals to communicate between the two of them. So they're not stuck together, even though they kind of look like they are, right? So this is called a synapse. This is where you are going to release neurotransmitters and communicate between two neurons. Now, just by looking at this figure, can you not, you should be able to tell which direction information is flowing, that direction. And now, just by looking at it, you should be able to tell me which one's the presynaptic neuron and which one's the postsynaptic neuron. Pre, post, right? Okay. Now, the synapse is where you are going to communicate between the two of them. At every one of these places, you're going to release neurotransmitters, which will then bind to receptors on the postsynaptic cell. Yeah? Quite often on the dendrites of the postsynaptic cell. Does that make sense? Okay, so now just by looking at this figure, you should be able to label a presynaptic axon and a postsynaptic axon. Yeah? And you can find one axon hillock on which cell? Postsynaptic, yes. You can find one cell body on the post. You can find dendrites on the post. You can find myelin sheath on both. I can't tell what kind of myelin it is, so you can't tell whether it's CNS or PNS in that figure, right? You can definitely tell direction of information flow, right? You can label axon telodendria on which one? Pre, right? And axon terminals on the pre, or synaptic knobs on the terminal, right? That makes sense? So just by looking at this figure, you can tell a tremendous amount of information about what's going on, right? That makes sense? Okay. All of that stuff ends up being stuff that, like, the faster you get it in phys, the easier the questions are, right? Okay. Which is why most people, Amy, take anatomy before phys. Crazy person. Okay. Oh, good. I'm glad. Okay. So you should be able to label all of that. Okay. FYI, there's more than one type of synapse. This synapse that you're seeing is neuron to neuron, and that's called a true synapse. But neurons can also communicate with muscles and glands, right? They can make things wiggle and spurt. And a neuron that communicates through a synapse with a gland is called a neuroglandular junction. And if it's neuron to muscle, it's neuromuscular junction. Now, the presynaptic cell on all of them is always a neuron, right? But the postsynaptic cell can differ. Does that make sense? Okay. Now, let's talk about our glial cells. Okay, so when you look at this figure, it looks like all the glial cells are the same. They hardly stain except the nucleus, right? They look all the same. They're not all the same. There's 6 different types of glial cells. You can't tell from a histology slide, but you can certainly tell from drawing what the glial cells are. Here is how to think about the glial cells, and I used this metaphor before, but I'm going to use it again. If you are talking about glial cells, the glial cells are way more abundant. Like there's 50 times as many glial cells as there are neurons, but they're much smaller, right? They end up making up half the brain volume, okay? And they're specialized in their functionality. And what they really are is the support staff for the neurons. Neurons are so highly specialized. All they really do is receive information, generate an electrical stimulus, and release neurotransmitters. That's really all they do. They don't know how to protect themselves. They don't know what they like to eat. They don't know when they're getting worn out. They don't know when they should take a break. They don't know how to do anything for themselves except this one specialized functionality. So the metaphor that I always use for neurons and their support staff is they have been movie stars since birth, and they don't know which side of the car the gas goes in, or how to access their bank accounts, or pay their cell phone bill, or anything. They don't know how to do a damn thing by themselves. So I want you to think of these two neurons like the Olsen twins, right? That's Mary Kate and Ashley up there. And so what they have is an entire support staff of people helping them to accomplish anything but their movie star lifestyle, right? Like anything except acting, although they don't act anymore. Now they're designers. So anything but their specific functionality is done by this support staff. And so when you're in the CNS, it's kind of like being in your LA home base. You got a big support staff. You got 4 different types of glial cells. When you go on location, you only take 2 with you, right? So the PNS has fewer glial cells. Okay, so now let's look at the glial cells and what they do. So you were in the CNS right here. You can tell it's part of the brain, right? You got Mary Kate and Ashley. And so their CNS support staff is 4 different types of glial cells. One of them you already met, which was their communication director, right? Which is the oligodendrocytes, which allows them to do communication. Does that make sense? Yeah? So we already met that, okay? The next one we're going to meet is the Astro Site, that big green one that you see there. Do you see why it's called an Astro Site? It looks like a star. Okay. They used to think the Astro Site was really just like holding stuff together physically, but now we figured out that it actually monitors access to the stars. What it does is there's a bunch of stuff going through your bloodstream that you do not want to get to your neurons, right? So what it does is it decides what gets out of that capillary into the nervous tissue. Does that make sense? So it's really monitoring communications, right? It's like whoever's in charge of their social media, right? Okay. So the astrocyte basically controls the permeability of the blood-brain barrier to protect the neuron, right? Really just protecting access. And then these microglial cells, those little bitty guys right there, okay, those are your security staff. And I know they look tiny, but they're freaking ninjas. They're little badasses. So what happens is within the central nervous system, you don't have any white blood cells in the central nervous system, so you need an internal security staff. What those microglial cells do is if anything funky gets into your CNS, they will whoop up on it, okay? So these are your security detail, right? Your bodyguards that you got. And then last one, and these are a little harder to conceptualize. Do you see this right here, these cells right here? Okay, you may or may not know, your brain and your spinal cord are both hollow. There are hollow spaces in your brain and your spinal cord. And so do you see those hollow spaces right there? Okay. So what you are doing in this picture is they're taking a chunk that gets you some of the tissue and some of the hollow, okay? So what these cells do, and they look kind of like epithelial cells, but they're not, okay? You don't have any epithelial tissue in the brain. What these cells are going to do is they are going to take all of the best stuff out of the bloodstream, meaning high levels of glucose, high levels of oxygen, high levels of all of the ions that you like in your brain. They're going to suck it out of there, and they're going to dump it into the brain as cerebrospinal fluid. So they make cerebrospinal fluid, and cerebrospinal fluid bathes the brain in the very, very best juice, inside and outside, right? So I couldn't come up with a metaphor, but in the last class, they're like, oh, that's your communication director. That's your person, no, they're PR, right? So I'm gonna take all the best stuff, right? I'm gonna say, oh my goodness, all this amazing stuff, and then I'm gonna dump it in here, and then I'm just going to bathe it over myself, right? So like Trump has security briefings, but he only wants you to bring good stuff about Trump. Did you hear that? He has security briefings in which he's like, do not bring me anything negative, only bring me cool stuff about me. Let's talk about me. Would you like to talk about me? Enough about me. What do you think about me? Okay, so these, don't tell me what you think about me. I don't want to know. Okay, so these are ependymal cells. And what these do is make cerebrospinal fluid. Good? Okay. All right. Now the Olsen twins are going to go on location, and they only get to take 2 of them with them. And the 2 that they take with them, if you ever have a neuron that's out of the central nervous system, it is really only going to have a personal assistant and somebody communication director. So if you are outside of the central nervous system, you get two kinds of glial cells. This one we already met, right? Which is what myelinates in the PNS, and that was called your neurolemocyte or Schwann cell, right? And then what happens is if you are a neuron and you're in the PNS, right, you're going to exist in a little blob called a ganglion. Sound familiar? And all you're going to have is one cell that is going to do all of the other functionality. So think of it like a personal assistant, right? It does everything. It tries to make sure that like takes out the trash, gets rid of waste products, make sure you have the proper amount of nutrients, does a little bit of protective functionality, right? And it's basically just doing everything for you, and it completely surrounds the cell body. Do you see it? And it's called the satellite cell. Can you see why it might be called the satellite cell? Because it's going all the way around the cell body, right? So now we have run into all 6 glial cells, right? 4 in the CNS and 2 in the PNS. That make sense? Anybody have any questions?
So which one's the second cell in the PNS?
The second one? Okay, this one right here. It's the one that does myelination in the PNS. So we already met it. It's the neurolemocyte, right? That make sense? Okay. All right, so before we leave today, let's talk about a couple of clinical applications of these kinds of things. So I think I told you guys back at the beginning that neurons are amitotic. Glial cells are not amitotic, okay? They can make more of themselves, okay? And for that reason, if you get a cancer in the central nervous system, is it more likely to be a glioma or a neuroma? A glioma, yes. So glial cells can still be cancerous. Now, is it impossible to get a neuroma? No, every damn thing that could go wrong will go wrong ultimately, right? So gliomas are far more common. And then the last thing is a disorder that is related to glial cells but manifests as a neuron problem. So what happens when your support staff leaves and you don't know how to do a damn thing for yourself? You lost your communication director. So what happens with MS or multiple sclerosis, it's an autoimmune disease. Your immune system makes a mistake and starts to attack the glial cells that form myelin sheaths, right? And what happens is you lose the capacity to send signals, especially long distances. Does that make sense? So it often follows a viral infection. You get a virus, your immune system gets all ramped up, and then it's like makes a mistake, and it starts to destroy your myelin sheaths. And so what happens is your nervous system short circuits. But do you know anything about MS? Where does it manifest first? In the periphery, where you depend the most on myelination, right? That's a long way to get a signal to go. So it manifests with loss of sensory and motor capacity in the periphery first, and then it can move in, right? So all you're doing is you're losing your capacity to send your communication, right? Does that make sense to you guys? So that's MS. So we will finish this set of notes on Monday. Oh, FYI, I don't know if I told you guys this already. I'm going to post a quiz for you that you can do online. You can do it open note. You can do it anytime before your test, right? It will have both the material that we have been covering and the material that we haven't yet covered online, right? And the reason I'm doing it online is simply because I don't want to quiz you while you're having a lab practical, and also I want the class time. I'm being greedy. Okay? Go ahead.
What's the difference between a Schwann cell and the oligodendrocytes?
Well, really the difference is how they form myelin. So this is the difference. Here's a Schwann cell. It devotes its entire self to the myelination. And this one, the cell body sits off to the side and does fingers, right? They end up accomplishing the same thing. FYI, the Schwann cell usually has a thicker layer of myelin. It usually makes it thicker. And Schwann cells, the last thing we're going to do next time in this set of notes is talk about how Schwann cells will help with healing damaged PNS tissue. So they behave very differently when damaged. So we'll talk about that next time. Cool? Okay, yes? MS or if it's also, I told them that it was like, for instance, the autocancer that follows tuberculosis. There was probably an infection, and it seems
Is that bad that I got happy on your side?
But I told her that I was gonna get it. Cause like she'll say something, I'm like, oh yeah. We need it.
Oh, yeah, and fibromyalgia is an autoimmune disease too, yeah.
So they
So there's a good hypothesis that has some, well, it's probably not a hypothesis.
And have to go back to 1050.
They think that when they get all autoimmune diseases,
Remember that sheriff said
in parts, because we quite often house another living being that is not us. Right? And so women usually get autoimmune diseases more than men, but not until they're in their 30s or 40s or 50s. Because they think that sometimes during the process of childbirth, although there should be no exchange between fetal and maternal blood, there is. Right?
Is that what MS?
Yes, exactly. You have male, we know that it is not an infertility carrier. It doesn't matter if it's a boy. So the way that they described it was because they found that there were XY cells in a woman, and they tracked that, and they were like, oh, these belong to your 26-year-old son. And then they said,
Wow.
they said, oh, well, now that violates 1985 if there wasn't any exchange. And since there is exchange, now can we figure out why women in their 30s, 40s, and 50s are way more likely to have autoimmune disorders than any other population group?
Because my grandma didn't get. I know we were rheumatoid arthritis.
Yeah. Yeah. A lot of times.
That's like one of my patients, she didn't have, like her MS, she was diagnosed at 14. She's a cheerleader, college cheerleader, husband was a college football player, and then as soon as she got pregnant, she's in a wheelchair now, can't walk, can't do anything.
So, they have an animal population actually has volunteered time and time and time again to be the control for, studies that are
I think that's
comparing
too
women
high.
who
I
have
think the
pregnancies
highest one
and
would be
women
like
who haven't. I mean, they're not all vessel virgins, but, because you don't have to be, right? But, I mean, you can be a nun.
I thought you had to be, so
No,
you just talking.
you can be a nun with kids. You just like have to.
I did not know that, and I'm
I thought you were
trying.
married to Jesus.
Well, you can't still be married.
Oh, okay, gotcha.
Oh, okay. So it's like your previous life before you accepted Jesus.
Before you accepted Catholic Jesus.
Right.
Because there's like a bunch of different Jesus. So before you accepted Catholic Jesus.
Yeah,
You'll see right here. They have volunteered time and again to be the control population for that. And they have a very low rate of autoimmune diseases. Yes. Wait, hold on. Somebody had a question.
We all do.
I accidentally stole this from my physiology lab. Can you return it? I just want to be honest. I'm just getting back to you.
Yeah, you'll be honest just a little later.
Wait a minute, they probably got you videotaping.
Yes.
So before we bomb with questions, what time do you have to leave?
Oh, I don't have to leave right this minute. I'm good.
Okay.